Hello friends. So today we'll be talking about the folate trap, which can also be referred to as methyl trap hypothesis. Now, vitamin B9, also called folic acid, or folate, and vitamin B12, called cobalamin, are both water-soluble vitamins. And the physiological roles of these two vitamins are related. That is to say, the absence of one vitamin will definitely affect the biological activity or the physiological role of the other vitamin. Now, the folate trap hypothesis or the methyl trap hypothesis can be explained on the basis that the absence of vitamin B12, that is cobalamin, will bring about a deficiency or an inhibition in the physiological role or biological activity of vitamin B9. So the absence of cobalamin will bring about an inhibition or a deficient activity or physiological role of vitamin B9. Folate can be obtained from our diet, but when it is obtained from the supplement or from supplements, it can be referred to as folic acid. So whether it is obtained from diet or from supplement, it plays similar role. When folate is obtained from the diet, during the digestive processes, give rise to polyglutamate. Polyglutamate. While if it is obtained from supplements, it is referred to as folic acid. And then during the digestive process, it gives rise to monoglutamate. The polyglutamate has to be hydrolyzed to form monoglutamate. So polyglutamate will undergo hydrolysis to form monoglutamate, and the enzyme responsible for such conversion is called the folate conjugase. So the folate conjugate, after digestion of this dietary folate to polyglutamate, folate conjugase will act on polyglutamate, hydrolyze it to form monoglutamate. Now, after the formation of monoglutamate, in the intestinal cells, monoglutamate, which is of course a form of folate, will undergo hydrogenation. So there will be addition of hydrogen at the position 7 carbon and position 8 carbon of the folate molecule, that's of the monoglutamate molecule, and that will give rise to 7,8 dihydrofolate. Okay, now after this there will be further hydrogenation. So this hydrogenation, of course, will take place in the presence of an enzyme called folate reductase. So there will be something like this. So folate will give rise to seven eight dihydrofolate, and then this will be catalyzed by folate reductase, then give rise to this. in the presence of vitamin C. So folate will undergo this process to form 7,8 dihydrofolate. Now this 7,8 dihydrofolate will also undergo a process of hydrogenation. So 7,8 dihydrofolate will also undergo hydrogenation to form 5,6,7,8 tetrahydrofolate. Catalyzed by folate reductase. Now, this 5,6,7,8 tetrahydrofolate, we can simply call it tetrahydrofolate, and we can write it in this format: tetrahydrofolate. Now, this is the active form of folate. So, folate in this form is not the active form. Folate in this form is not the active form. Whereas when it undergoes the steps of hydrogenation, it becomes active. So this is the active form. This is the biologically active form. So this folate carries out its physiological role in this form. Now, this still occurs in the intestine. But before it can be absorbed into the circulation, tetrahydrofolate will have to combine with methyl group. So methyl group will be attached to the position 5 of the folate molecule, and that will give rise to 5-methyl tetrahydrofolate. Remember, I said let this represent tetrahydrofolate. So a methyl group will be attached to the 5th carbon position, okay, of the folate molecule, and that will give rise to 5-methyl tetrahydrofolate. Now, the absorption of folate will occur in this form. It is at this point that this will be absorbed. Of course, the absorption of this is also an active process, okay? So it can be absorbed with the help of a specific carrier protein. When 5-methyl tetrahydrofolate has been absorbed into the circulation, it will undergo a process of dissociation. Of course, it will dissociate to form a methyl group, and then it will give rise to tetrahydrofolate. Remember, I stated earlier that the active form of the biologically active form of folate is tetrahydrofolate, and that before it can be absorbed into the circulation, it must combine with a methyl group. But then after being absorbed into the circulation, it will dissociate to release the tetrahydrofolate from the methyl group. Now, it is a tetrahydrofolate that will go on to carry out the physiological role of folate. So in the circulation, this will undergo dissociation to form tetrahydrofolate. But that formation must occur in the presence of cobalamin. Cobalamin, vitamin B12. So this dissociation must occur in the presence of cobalamin. So in the absence of cobalamin, this dissociation cannot occur. So in the presence of cobalamin, 5-methyl tetrahydrofolate will give rise to methylcobalamin. Of course, it's going to combine with this methylcobalamin and tetrahydrofolate. Now, this tetrahydrofolate plays a role. It goes and plays a role. Okay, it functions in what we call the 1-carbon metabolism. one carbon metabolism. One carbon metabolism. So one carbon metabolism is simply a complex series of biochemical reactions that has to do with the transfer of one carbon unit, okay, between molecules. There's a transfer of one carbon unit between molecules. Now the one carbon unit could be in form of a methyl group, it could be in form of a methyl group or a formyl group, right? So tetrahydrofolate plays a role in transferring one carbon unit, right, between molecules, and that can lead to a lot of physiological functions such as DNA synthesis, RNA synthesis, and several others. One carbon metabolism can lead to the synthesis of purines and pyrimidines, which are building blocks, okay, for DNA and RNA synthesis. It also plays a role in methylation, methylation of RNA, methylation of proteins, methylation of DNA, and several others. There are several functions, several physiological roles. So all these physiological roles occur through the process of one carbon metabolism enhanced by tetrahydrofolate. Now methylcobalamin will then combine with homocysteine in the body, okay. Methylcobalamin will combine with homocysteine in the body, and then it will give rise to the combination of methylcobalamin and homocysteine will give rise to methionine and cobalamin. So you see, methyl will be attached to homocysteine to form methionine, and then cobalamin will be free back into the circulation to continue the process from here. Now, this methionine that is formed is a precursor to S-adenosyl methionine. S-adenosyl methionine. Now, S-adenosyl methionine also plays a role, okay? This plays a role in donating methyl group. Remember, this methyl group was transferred to this to form this. So it has methyl group in it. So what this function is that it donates or it transfers methyl group to several molecules. The methyl group could be transferred to RNA, DNA, proteins, and etc. That is to say, it plays a role in methylation. Now, methylation is basically a process where a methyl group is attached to a molecule, and that attachment will affect the structure and function of that molecule. For example, DNA methylation. In DNA methylation, a methyl group is attached to the DNA molecule, and that helps DNA, okay, to regulate gene expression. So at that point, the DNA will determine the gene that will be switched on or the gene that will be switched off. So that role or that physiological role of regulating the gene to know which one is switched off and what is switched on is called DNA methylation. And that DNA methylation is possible due to the presence of S-adenosyl methionine. Now, this is our topic, folate trap hypothesis. Folate trap. Now, assuming cobalamin is deficient, cobalamin is not there. So let's say cobalamin is not there, okay? Now, in the absence of cobalamin, what is going to happen is this molecule cannot dissociate to form this, okay? So this molecule cannot dissociate in the absence of cobalamin to form tetrahydrofolate, which of course is the biologically active form of folate that carries out the physiological role of folate. And when this cannot be dissociated, it means The biologically active form of folate has been trapped in this form. So this will be in the circulation, but it cannot function. That is due to the absence of cobalamin. So a process or a condition where there is deficiency of cobalamin such that 5-methyl tetrahydrofolate cannot dissociate to form tetrahydrofolate. At that point, the tetrahydrofolate, which is a biologically active form of folate, will be trapped in this form. It will be in the circulation, but it cannot function. So these processes explain the folate trap or the methyl trap hypothesis. Apart from the fact that folate has been trapped, methyl has also been trapped. And from what I stated there, the methyl group that was released there to this got attached to this to form this. And I also stated that S-adenosine is involved in donating or giving out methyl group, which I gave an example of DNA methylation. So when this cannot dissociate, also the methyl, the methyl group will also be trapped. That's what we call the methyl trap hypothesis. So both the methyl and the folate will be trapped. Now, when the biologically active form of folate is being trapped, like I stated earlier, this molecule or this compound will be in the circulation, but it cannot function. So when folate in the circulation cannot function, it will result in what? Folate deficiency. So what it means is the absence or the deficiency of cobalamin will result in deficiency of folate.